Welcome back. I'm Dr. Muskan, and in this video, we will be continuing our discussion on viral infections. In this video, I am going to talk about hepatitis. Now, what is hepatitis? Hepatitis means inflammation of the liver. What is viral hepatitis? Viral hepatitis means that there is infection and inflammation of the liver due to viruses. There are a lot of viruses that can cause viral hepatitis, which includes hepatitis A virus, B virus, C virus, D virus, and E virus. We will be mainly discussing about hepatitis B and hepatitis C virus. Let us continue with hepatitis B. I hope you remember the subheadings which we are going to discuss. Now, the family of hepatitis B. It belongs to the family of Hepadnaviridae. It belongs to the family of Hepadnaviridae. Now, what is the structure of the hepatitis B virus? It contains double-stranded, partially circular DNA and also has an envelope. Now, what does this mean by partially circular? Normally, we have circular DNA which is complete, but in hepatitis B, we have one strand which is very complete and the other strand is partially complete. This is called as partially circular double-stranded DNA, also called as relaxed circular double-stranded DNA. Now, if we look into this DNA, we will have genes. Now, what are these genes? S gene. It codes for HBsAg. Now, what is HBsAg? It is hepatitis B surface antigen. Then we have the C gene which codes for hepatitis CAG. Now, what is hepatitis CAG? Hepatitis B core antigen. Now, if I draw the structure, you will be able to see that there is double-stranded partially circular DNA at the center. Then it is surrounded by the protein capsid, followed by which there will be an envelope. This is the envelope. So, we have, when you look into the envelope, you will be able to see the surface antigen HBsAg. And when you look in the center, you will be able to see the core antigen HBcAg. We also have the pre-C gene that codes for HBeAg. Now, what is HBeAg stands for? Hepatitis B envelope antigen. So, here we also have HBeAg. Now, this particle which I have drawn with the double-stranded partially circular DNA with protein capsid and envelope. This all together is called as the Dane particle. The Dane particle. Now, we also have P gene. This P gene codes for polymerase enzyme. Now, what does this polymerase enzyme do? This polymerase enzyme has three activities. It acts as DNA polymerase. It has reverse transcriptase activity. It also has RNase activity. So, these are the three activities by the enzyme polymerase which is coded by P antigen. Then we have the last gene, the X gene, which codes for HBxAg, and this is the regulator protein. This is the structure of hepatitis B virus. Hepatitis B virus has a special property that it has partially circular double-stranded DNA where one strand is complete and the other strand is incomplete, and it has genes coding for various proteins HBsAg, HBxAg, HBeAg, HBcAg, and polymerase enzyme. Now, what is the mode of transmission? Hepatitis B virus has various modes of transmission. That means it is going to enter in our body through various routes. Through the heterosexual route, through blood transfusion, via needle stick injuries, via IV drug abuse, and also hepatitis B virus can get transmitted from mother to the baby via the perinatal or vertical transmission. So we also have the perinatal transmission. Now, via this various modes of transmission, the virus is going to enter in our body. Okay, now once the virus enters our body, it is going to reach the liver because it is the hepatitis virus. So it's going to reach the liver. In the liver, we have the hepatocytes. These hepatocytes are nothing but the cells of the liver. In the hepatocytes, this is the hepatitis virus. It reaches the hepatocytes. So we have the hepatitis B virus and we have the hepatocyte. Now on this hepatocytes, we have a receptor. Now what is this receptor called as? This is called as sodium taurocolate co-receptor. To this receptor, this hepatitis B virus is going to attach. So, hepatitis B virus will attach to the hepatocytes via this sodium taurocolate co-receptor and enters the hepatocytes. It enters inside the hepatocytes. Once it has entered into the hepatocyte, it is going to undergo uncoating. That means the envelope will be lost and the partially circular double-stranded DNA is now going to enter the nucleus of the hepatocytes. It is now going to enter the nucleus of the hepatocytes. So, if I draw a diagram of a hepatocyte, this is the nucleus of the hepatocyte and we have this partially circular double-stranded DNA which is going to enter the hepatocyte. This is the genome of our own cell, that is the hepatocyte, and this is the partially circular double-stranded DNA. Once it enters the nucleus, it is going to undergo replication. Now what is going to happen is this partially circular double-stranded DNA is now going to become completely circular double-stranded DNA. This is how it is going to complete its structure. So we have the partially circular double-stranded DNA which becomes completely circular double-stranded DNA. Now this DNA is going to act as a template for the transcription. Now what is transcription? Transcription means that the DNA is going to get converted into RNA. So, we have three types of mRNAs that are going to be formed. What are these three mRNAs? We have the pre-core mRNA, we have the subgenomic mRNA, and then at the last we have the pregenomic mRNA. Pregenomic mRNA. Now, what does pre-core mRNA translate into? Now, what is translation? Translation means that it is going to form the proteins. Now, pre-core mRNA is going to translate into HBeAg. We already know what is HBeAg. It is hepatitis B envelope antigen. Subgenomic mRNA, it is going to form two proteins. What are those two proteins? We have the HBsAg, which is the surface antigen, and we have the HBxAg, which is our regulator protein. What about pre-genomic mRNA? It is going to form HBCAG, which is our core antigen. It is going to form the DNA, which is the polymerase enzyme. And finally, we have a mRNA, which is pgRNA. Now, what is this pgRNA going to form? This pgRNA is going to undergo reverse transcription. We already know what is reverse transcription. Reverse transcription means that the RNA is going to get converted into DNA. So, this pgRNA is now going to convert into a partially circular DNA. This is how it is going to form the DNA and the viron particles, and these viron particles are going to come out of the hepatocytes and continue the further cycle. So you can see that how hepatocytes are getting affected by the hepatitis B virus.